Hi, we're going to talk today about nucleic acid components and structure. So the objectives will be to identify the components of a DNA molecule, compare different nucleic acid molecules, and identify the components and levels of compaction of the DNA. So you're all very well aware of the central dogma, right? We have a double-stranded DNA that then needs to be transcribed. So this DNA is going to be kind of the basis for the information that will be passed on from generation to generation. So this DNA must be well protected, maintained, and passed on. So the DNA will then be transcribed in order to pass on part of the message, which this message which is made of RNA, will be passed on through translation to become proteins. So we're going to be talking about these nucleotides in particular right now and the whole structure of the double-stranded DNA. So a nucleotide is actually composed of three items: a nitrogenous base, which will be determined, as you well know, as A, T, C, or G, a sugar, which in the case of DNA is a deoxyribose. In particular, it is the 2' deoxyribose. We'll see how that differs with the RNA, the ribonucleotides. And it's also composed of a phosphate. You can see that the phosphate has lots of oxygens, which is actually charged. So this whole phosphate will provide the negatively charged component of the whole DNA. So this is a nucleotide. Again, a nucleotide is going to be composed of a nitrogenous base, a sugar, and a phosphate. There are very important items in this nucleotide. We do not expect that you know the whole chemical structure of each of these components, but there are a few things that you must know because they will help you understand better how replication happens and things like that. So the first thing is that the ribose has 5 carbons that are labeled from 1' through 5'. The 1' carbon is covalently bound to the nitrogenous base. The carbon at 2' in DNA is a deoxy, meaning that it has no oxy, right? The oxy aspect of it is not there. The 3' has an hydroxyl function, which is going to be extremely important both for DNA and RNA to allow for incorporation of more nucleotides in the strand of nucleic acid. And then the 5' end is the one that is covalently bound to the phosphate. So again, we have the nitrogenous base. Once a nitrogenous base is bound to a ribose or a deoxyribose, now this is called a nucleoside. And when we have the actual 3 components, the nitrogenous base, the sugar, and the phosphate, the negatively charged phosphate, now we're going to have a nucleotide. Differences between DNA and RNA. So one of the most important ones is through the name, right? The DNA is called the deoxyribonucleic acid. Deoxy meaning that it lost its hydroxyl function at the 2'. In the case of RNA, it's a ribonucleic acid. You can see here this is an actual ribose, and so it still has the hydroxyl function at the 2' end. Otherwise, as you well know also, DNA has a thymine while RNA will have uracil. I've mentioned before, right, that the names of nucleotides, nucleosides, and nitrogenous bases differ depending on whether or not the ribose is bound and or they have 1, 2, or 3 phosphates bound to it. As an example here, I am showing you the uridine triphosphate. So this is a uridine bound to its ribose, and as you can see, there is the 2' OH for the RNA, and then it's triphosphates because you have 1, 2, and 3 phosphates bound to it. And here we have the thymidine triphosphate because, so it is part of the DNA. It does not have the 2' OH group, right? And it has 3 phosphates. When you think of a nucleotide, usually the nucleotide that is inside the DNA will only have 1 phosphate. These will be gone, and we'll see how that happens in the replication process. Okay, so we talked about a nucleotide. Now we're going to start talking about, how this nucleotide is the building block of the double-stranded DNA. So a strand of DNA, I will always talk of a strand of DNA from 5' to 3', okay? And again, we've mentioned that the 5' is the carbon that is covalently bound to the phosphate. So as a general rule, convention, phosphate will always be on the 5' end of the DNA, and we will start with a 5' end, and then you're going to have phosphate deoxyribose, phosphate deoxyribose, phosphate deoxyribose, phosphate deoxyribose. And it's going to end with the 3' hydroxyl group that is going to be free for the next nucleotide to come and be able to bind at this location. We'll see that with the replication process. So in the end, the DNA is covalently bound through this backbone of phosphates and sugar. The nitrogenous bases will be piled up, but will not be covalently bound to each other, right? They are just covalently bound to the sugar. Here we have the 4 nitrogenous bases of the DNA: thymine, adenine, cytosine, and guanine. At this point, you should be able to complete nucleic acid 1. We're going to talk now, so we talked about one nucleotide, then we talked about, multiple nucleotides form one strand of DNA. Now we're going to talk about this idea of the double-stranded DNA. You're probably very familiar with this idea that double-stranded DNA forms a double helix. Often this double helix is considered a right-hand double helix because you can imagine how if you were to grab it and follow the ridges, your right hand would be going up. So that's what we call a right-handed double helix. You can see how, again, 5' end with phosphate sugar, etc., up all the way to the 3' end. We have the nitrogenous bases here of the left-hand side strand. And then, opposite to it, right, we're going to have an anti-parallel strand going from the 5' end, but now this 5' end is kind of at the bottom, phosphate ribose, etc., to the 3' end. These nitrogenous bases, again, are covalently bound to the ribose, but that's it, right? The other bonds that they are able to form now are either van der Waals bonds or hydrogen bonds, right? Meaning that they're going to be able to form these loose bonds, but they are strong enough that they allow the bases to kind of maintain each other there. So you can see here the depiction of these double helix, and one thing you can notice actually is that there is a major groove right here, and right here, and there is a smaller groove right opposite to the major groove, which is going to be called the minor groove. This is going to be important in the interactions with the proteins, for example, or interactions with other molecules. In particular, here we have the image of Dr. Rosalind Franklin, who was the person who obtained the image of the X-ray image of the crystallized DNA, which allowed for the confirmation of this double-stranded double helix structure. So I was talking about this idea of the major groove and the minor groove, and you can see how in the major groove the bases are a lot more exposed than in the case of the minor groove, where it's very hard to kind of imagine being able to interact with any of the bases in a specific way. So what this determines actually is that the major groove is actually often associated with specific interactions between the protein and the DNA, because then now the proteins, enzymes can actually identify a specific sequence and then bind particularly to this region because of this sequence. The other aspect also is that proteins are often quite big, and so in order to kind of fit in properly, they have a better kind of anchorage right here than in the minor groove. So the major groove is often associated with interactions with the proteins. So there is one notion of the DNA that comes back often, and this is the idea of the double-stranded DNA. In order for DNA to be double-stranded, it must be reverse and complementary. We're going to take a look at those two items. So reverse meaning it's anti-parallel, right? And so this again goes with the idea we start with one strand at 5' end to the 3' end, and the opposite strand will be going on the opposite direction from the 5' end to the 3' end. Now the idea of the complementarity comes with this idea of canonical base pairing. You are probably very familiar with this also. A and T will always be together, G and C will always be together. In RNA, you would have A and U that will always be together. Now, why is that the case? Well, when you look at the chemical structure of these nucleotides, or nitrogenous bases rather, what you can see is that adenine and guanine are actually called purines, and they have 2 cycles, so they're pretty big, while thymine, cytosine, and uracil in the case of RNA are pyrimidines, and these have only 1 cycle. So what happens is that when you have them side by side, you can see how the 2 strands can maintain a distance that doesn't vary much, because in both cases you will always have a purine associated with a pyrimidine, purine with a pyrimidine, purine with a pyrimidine, okay? Just as a reminder, one way that it's easy to remember is that a pyrimidine, which has a PY, is associated with the nitrogenous bases that have a Y, thymine and cytosine. And once you know that uracil is also similar to the thymine, then you can put it also in the block of the pyrimidines. At this point, you should be able to complete nucleic acid 2. We're going to talk now about the DNA levels of organization. What you need to understand is that actually chromosome 1, for example, of humans has 249 million base pairs, and that 10 base pairs, right, measures 3.4 nanometers. All of these numbers might not mean much to you, but I would like to ask you, how long is chromosome 1, right? So try to see how you can figure out to determine how long chromosome 1 is. And just to give you an idea, a nucleus of a cell is usually, 10 to 5 micrometers. So we're going to talk in class about an analogy of how hard it would be to fit in, right, a string that is that long in a space that is this small. And so in order for that to happen, it is very important that the DNA is super, super, super organized, and for that, then there's a structure that is needed. And so there's complexity in this organization. So we talked about the fact that nucleotides are the building blocks of the DNA, right? So once nucleotides are put together, we're going to have our double helix, double-stranded DNA. Now, this double-stranded DNA has to be compacted. So you might remember we talked about phosphate being negatively charged. So because phosphate is negatively charged, it actually helps these histones to bind to it. So this should tell you pretty easily what charge do the histones probably carry. So these histones the DNA wraps around the histone. So we already have, another level of organization here. Now these histones often form tetrads, so they come into 4s, and so this is another level of organization. So for each histone that has some DNA wrapped around, now we're going to have more histones that kind of come together and wrap around each other. And so that's what we call a nucleosome. Now these nucleosomes are going to start wrapping around each other even more, and that's going to be the chromatin fiber. So this chromatin is considered often a loosely structured DNA. It's often the place where it's going to be possible for the DNA to initiate replication or to start with transcription. Now this chromatin has to be even more structured, and so it's going to start forming these loops. And again, these loops are going to be even more compacted. They're going to form this chromatid. And as you know, two chromatids will form one chromosome. So there you got it. We have a chromosome. So you see how there's really a stepwise hierarchy to organize this DNA so that this very, very long strand can fit in this very small nucleus. Also remember that the DNA is not, the human DNA is not just composed of one chromosome 1, it's two chromosomes 1, but it's actually also another 22 pairs of chromosomes. So there's really a lot of DNA that has to fit in this very, very small nucleus for the case of humans, but it's very similar for the case of other living organisms. So at this point, you should be able to complete chromosome structure 1. So take-home messages: DNA is the genetic material for all living organisms, right? It is the basis for messages to be passed on. With the small exception of viruses, that some of them use RNA. A few viruses use RNA as their genetic material. The DNA differs from RNA. We talked about the 2' position, where in the RNA is a hydroxyl function, but in the DNA is deoxy, right? So there is no hydroxyl function at the 2' position. Also, DNA has thymine, while RNA will have uracils. The nucleic acids form reverse complementary double-stranded helices, right? So the DNA is most often found as double-stranded, while the RNA is often found as single-stranded, but can also form double strands. So mRNA is often found as single strand, but tRNA is one single molecule of RNA that is folded, and in that folding, it becomes double-stranded. And then we've also mentioned, right, the 3' position of the ribose, which is very important to keep adding nucleotides, but we'll see that in the replication process. The 5' position of the ribose, which is bound to the negatively charged phosphate, and the 1' position of the ribose, which is covalently linked to the nitrogenous base. Thank you.